Okay, so starting off from where I left off, so let's talk about polymers and entropy, and then we'll see how polymers and entropy is being used to understand the self-organization of chromosomes within the bacterial cell. I'll give experimental details also. So the broad problem was, as I told you, in bacterial cells, there's no specific machinery, dedicated machinery like the mitotic spindle, which segregates the two bacterial DNA. In the E. coli cell, there's only just one DNA to segregate the two daughter DNAs to two halves of the cell along the cylinder. So, with that background, with that in mind, we shall discuss some physics. Okay, so switching the mode from biology to physics. Now, what you know is if you have a box with two kinds of particles or even two identical particles separated by a wall, right? So this has some blue particles, and this has some red particles. And they might even be oxygen molecules. They're just marked different so that they are made distinguishable to the eye. And if you remove the wall, the basically molecules are going to mix. You see, so here, I mean, this is just the red and blue colors are so that they can identify. You see, they have mixed inside the box, and the reason that they mix is to maximize their entropy. If the wall is not there, then each of the blue particles can explore all the positions within the entire box, and similarly so for the red particles. And that will essentially uh, increase the number of microstates. In case you have done your stat mech, then it will have access to more larger number of microstates. A microstate is defined by the position of all particles and the velocity of all particles in classical statistical mechanics. And larger the number of microstates, higher the entropy. So gas molecules mix. Now, however, in contrast to that, if you have ring polymers are long polymer chain-like molecules whose ends are connected. And if you have two ring polymers mixed in a cylinder, again here blue is one chain and red is another chain. And assume the only interaction between the chains is the monomers of the chain cannot overlap. And I will discuss polymer chains a bit more and what is entropy, so on. If you start off with that mixed configuration or overlapping configuration, except instead of molecules, you have a connected chain. You have two ring polymers, and the colors are just so that you can distinguish between them. Then, and if it's under high confinement, confinement is extremely important, then they would spontaneously demix into two halves of the cylinder. You see that there is a cylinder. This won't happen in a box. So this asymmetry of the cylinder is important. They would spontaneously demix. So, and this is the property, comes from the statistical physics, and they would demix because of entropy. Let me show this before going on. So this is a schematic of a ring polymer. This is just bead spring model. You have beads connected by springs. The only interaction is that two beads cannot sit on top of each other. So suppose there are beads of two polymer chains, right? Then they can't. Uh, I mean, so basically, you have excluded volume. Two particles cannot sit on top of each other, electron repulsion or whatever you want to say. Uh, and this is a ring polymer. The ends are connected. Suppose there are 100 monomers, or I think in the movie there are 200 monomers in the chain. And what you have initially, I'll stop the video. The red and blue are two chains identical. They have no attraction. The only interaction is the chain connectivity, and two particles cannot sit on top of each other, excluded volume interaction. They have been, I mean, we have taken special care to start them off in the mixed state. So blue is one chain identified by connectivity, and red is another chain. All that you have is repulsive interaction among each of the monomers. So the blue, so this monomer has the same repulsive interaction with this red monomer as well as this blue monomer if it's on top of each other. There's no other way you can distinguish individually. But chain connectivity is different. And if they start out mixed, you will see automatically ring polymers demix and they go to two halves of the cylinder. What is important is that this confinement is significant. It's whatever its spontaneous size going to be, this confinement is significant. We'll talk about this. See, one half is blue, one half is red. They spontaneously demix, right? So just looking at the previous slide. So gas molecules mix, but ring polymers demix in a high confinement in a cylinder. And this was suggested in 2006 by Sujun and Bellamunda and so on and so forth in 2000. So they observed that for a normal statistical physics ring polymer chain. And they suggested maybe that bacterial chromosomes, which are also ring polymer, the driving force for their segregation in the absence of any active energy consuming driving machinery might simply be entropy. Take two ring polymers, they would spontaneously segregate. Bacterial chromosomes, E.coli, are ring polymers. Maybe they segregate due to entropy because no method was known. It's still, even of today, it is an open problem because when they were suggested, obviously, biologists have their favorite proteins, but so they were not pleased. I mean, there's so much must be happening, but they're just saying due to entropy. But by 2010, they were writing Nature Review microbiology articles saying that doing a much proper study than the 2006 work, mentioning that, it has the right concentration and aspect ratio, blah, blah, so that entropy could be a driving mechanism. And by 2013, others were writing that most probably entropy is a major key component of the segregation of chromosomes. It might not be the whole story because timescales, etc., do their maths, and there were many open questions. Even now, there are many open questions. Later, we come in and contribute to this area. So even today, if you read review papers, you'll say, yeah, entropy seems to be a primary key component which drives segregation of chromosomes, but certain proteins are also important. What is their role? That is not understood. Maybe entropy may be half the story, but maybe not the full story. So two ring polymers, how are they related to chromosomes? Basically, bacterial, the mother DNA gives rise to two daughter DNAs. Here, what the movie I showed you, it's full two ring polymers. There's nothing biological about it, right? But in the bacterial cell, actually replication and segregation goes on in parallel. So as the mother is undergoing replication, they also undergo segregation. It doesn't really start with this full ring polymers, but we'll come to that. So what is the point? The point is entropic segregation to two halves of the cell as replication is in progress. Some of it was also modeled by Suk-Jun, who he later got so interested in this problem, he started doing experiments. He does experiments in San Diego. You can look up his webpage and his journey moving from a theoretical physicist to this computer modeler to an experimentalist. It's quite interesting. And this is the current status of the field when I entered. Okay. So here, before I go further and talk about chromosomes, I want to talk a bit about polymers because I understand there might be many physics students who might not be uh, comfortable with this idea. And what does it mean that entropy segregates ring polymers to two halves of the cylinder? Why do ring polymers should demix? Let's talk about the physics of this. First, polymers. So typically, you have been introduced to polymers in such hydrocarbon chains and so on and so forth, where each is a monomer and these are repeating units. The way we first look at polymers is typically the spherical cow. So we replace all the chemical details by beads, where we say, okay, we are looking at only those properties which are statistically universal across all long chain molecules. So we are really not looking what does polythene do and what does polystyrene do. We really are not capable of doing that. Chemistry is important if you want to make a particular material, the phone cover or something, and if you replace polystyrene by polythene by some other long chain molecule, you're going to have a massacre. So for some problems, for some issues, for some technology, chemistry is important, but one can also look at polymers as the statistical physics of very long chains, and we're looking at only universal properties, statistical physics properties, which are independent and universal for all long chain molecules. So we can't look at what temperature does this polymer melt and at what temperature is its glass transition. No, for that we need the chemistry details. But we are not looking at those. Let me explain. So this is what I have said. Chemical detail of each monomer is unimportant for some of the statistical physics properties that we are looking at. So we model essentially now large dynamics comes into the play. So we look at polymers like this. So there's a background fluid which we have not explicitly modeled, but it's the effect of the background fluid is being modeled by the Lajma equation for each of the beads, and each of the beads spheres are connected by springs, which again are not shown. I mean, you essentially have a spring potential, and what you see as each of the monomers are undergoing random kicks, they want to do Brownian motion, but they're connected along the chain, as a consequence of which you see that the polymer is constantly changing conformation, configuration. So it's not only that the individual monomers are doing Brownian motion, the whole chain itself is also doing Brownian motion. The center of mass of the chain is also moving, is also diffusing. Yeah, I can use now the word diffusing, but moreover, the internal degrees of freedom, this is the conformations, the angles of different vectors which joins one beat to the other. The angle between adjoining vectors are also changing. So they're changing conformations, they're changing configurations. And if they are changing their configurations, what also happens, so here you have similar videos. So here you have 100 monomer chain, which was initially compact, and you see that it is again spread out, and it's again changing conformations and configurations. As a consequence of which the actual energy of interaction between monomer and monomer, they hardly touch each other. It's only if they touch each other, there's a repulsive interaction and they're not allowed to overlap, right? It's rather weak. But this polymer has a large number of microstates, large number of configurations, large number of conformations is related to large number of microstates. And this large number of microstates is related to entropy. So this polymer has a huge amount of entropy. So F equal to U minus free energy F is equal to U minus Ts. The internal energy contribution is very minimal. At thermal KBT, these harmonic springs are nearly of unit length. They fluctuate a bit, but I mean, kappa is high, but the energy of interaction is only repulsive interaction, so that beads don't see each other. That's a very minimal, but they have a huge amount of entropy, and you see that they're constantly changing conformations due to uh, fluid molecules bombarding it, Brownian motion, which is now modeled by the Lashma equation. We have not explicitly modeled all of the fluid molecules. Now here, what I want to show is essentially you have some chains which are semi-flexible. This was a flexible chain. And I already told you the statistical physics starts from a random walk. So it has an average size, which is much smaller than what if the polymer was stretched out. So Each bond can point in any direction. However, here is a semi-flexible. There's a energy cost for bending. The consequence, you will see that these chains are not bending as freely as these were. Locally, they are straight, but over large distances, they look flexible, right? And there are four chains. They are colored in different colors. So in this case, the equilibrium configuration is not just entropy driven, but here energy also plays a role. If it could bend a lot, then it would have much more number of microstates. Yes, entropy would be high, but that would also lead to a penalization, high value of U. So it is the free energy. So this is in contact with the thermal bath, for those who know. So it's a canonical ensemble. So here entropy is not maximized, but free energy is minimized. And here both energy and entropy is playing a role. So number of conformations taken is significantly less compared to the number of conformations taken here, because conformations with high bends would lead to high values of energy, which would increase the free energy. For such a system, in canonical system, in contact with a heat bath, where the temperature is defined, free energy is minimized. So these two are balancing itself. So its energy is trying to minimize, and within that, I mean, not that energy is minimized, the combination of these two gets minimized to for the equilibrium. Okay, so that's the idea of entropy. Number of microstates, number of configurations. That's the idea which you want to say. And I'll come back to the ring polymers. Here you have essentially computed a simulation of a ring polymer, four ring polymers actually, and they were compacted. You see these red lines because there is some periodic boundary conditions. So this chain can be seen here as well, and so on and so forth. What you see is within the limit. So now it has been constrained. The two ends are together, right? They try to avoid each other is one thing which you could see uh, compared to this case. Right, and within that, within the constraint of the two ends should be together, they are again taking large number of conformations, four ring polymers. So that's, and again these number of higher the number of configurations, higher the entropy. But here they are constrained that the two ends must be joined. So of course there's fewer number of conformations that it can take. Of course, this has larger number of monomers than this also. So it tries within the constraints, it tries to maximize its entropy. There's no free energy cost as in the semi-flexible case, right? So from there, let's explore the idea of entropy a bit more. Now, if you take a polymer chain, if it's completely compacted, you see it tries to spread. It spreads, and each yeah, it spreads in a way so that it can take the largest number of conformations. Now, if you completely stretch a polymer, you constrain it so that so it's a new equilibrium where the two ends are held fixed together. Then completely stretch, the number of conformations it can take is only one, right? That's only one microstate, very low entropy. Leave it, I mean, there's no energy cost. Suppose if you just stretched it, not stress the springs, just stretch to make it straight, just release it, it will come back and become a coil. From stretch state, it will become a coil. Why will it become a coil? Because the number of conformations it can take, the number of ways these bonds can arrange themselves is the maximum when it is not fully compact, but some average size. Remember, yesterday when I was talking about random walks, again, when the two ends were close together, when the end of the random walk was close to zero, That's the case where you could take the largest number of random walks. And remember, at the end, I told that each of these vectors of a polymer along the bond vectors, could be thought of as a random walk in 3D. So again, the question is, under what conformation, under what condition would it be able to take the largest number of conformations? Largest number of ways can it arrange itself? The same question which we were discussing yesterday in a completely different context. So much of the physics of random walks can actually be transferred to the physics of polymers, except this is a self-avoiding random walk, if you know what I mean from the previous class. Okay, so here I want to again tell you that a polymer, a rubber band, is essentially a polymeric object. So more complicated, the multiple polymers connected by gels. When you pull, you can stretch it easily. It's much easily than you can stretch you can stretch a steel rod. And the reason is, in a steel rod, the Young's modulus or the elastic modulus, gigapascals, right? In and you need a huge, a human can't even stretch it. That's because when you stretch it, all the energy of interaction in the steel rod is essentially electronic in origin. So the atoms are held together by electronic interactions. I mean, the atoms, electrons overlapping with each other, and those. So the energy of interaction is electron volts. That's 100 times more than the thermal energy. In the polymer, what you have when you stretch it, there's not much of energetic interaction. What you're working around is just stretching a polymer chain like this. Hence, its entropy is decreasing. Those energy costs are order of kBt, much weaker than the electronic energy, electronic interaction energies. Hence, when you pull it, you can, I mean, basically the force that you are applying or the restoring force is entropic in origin. Why should you believe me? See, if you take a steel rod and you take it to a higher temperature, it's going to become soft. Means you can pull it relatively easy so that it doesn't melt. You heat it. Yeah, that you might have an intuition of. If you take a rubber band and take it to a higher temperature so that you don't burn it, then you will find to pull it by the same amount, you will need a higher force. So this is in marked contrast in how it works for that of a metal rod. Okay. And the reason that this is so different, so at higher temperatures, you need higher forces to pull a rubber band, a polymeric material, by the same amount, whereas for a steel rod, where the underlying interaction is electronic in energy, at higher temperature, you need less force. In rubber band, you need more force to pull by the same amount. I mean, you're not pulling the steel and the rubber by the same amount. There you will be pulling by millimeters. Here you can pull it double the length. But rubber band, making it double the length at higher temperature, you will have to apply more force because now you are working against entropy, and entropy comes. So you're decreasing the same number of microstates, but the change in free energy at a higher temperature on pulling it, on stretching it is more because there's a T sitting here. U is hardly contributing anyway. I mean, this is very minimal contribution for a rubber band. If you stress the same amount, the number of microstates which is getting decreased is the same. Okay, which is basically contributing to S. But since T is higher, the change in free energy for the same change in number of microstates which contributes to S is more. Change in free energy is more. So the force that you are working is not dominated by del U del X, which is the force, conservative force, but it is del F del X, and it comes with an increase, uh, yeah, increase in, number of microstates decreases. Yeah. The change in, so hence its free energy contribution on stretching number of microstates decreases, as a consequence of which free energy increases because it's coming with a minus sign. If entropy decreases with the minus sign, free energy increases. And the system would try to decrease its free energy. Hence, it's this del F del X. Restoring forces are entropic in origin. And any material whose Young's modulus increases or increase of temperature, you could guess that underlying forces, the dominant forces compared to UNTS, more entropic in origin. As if it's energetic in origin, its Young's modulus or elastic modulus will decrease on increase of temperature. So that's another important, so some understanding of entropy. Now, what is said here long back, that ring polymers segregate to increase its entropy. How would segregation lead to increase in entropy? So I had to give you this concept of entropy before I discuss this. Right, so now let's discuss this. Uh, so what happens is, if you have an infinite cylinder, and you have two polymers, let's think of it in 2D and you can extend the idea in 3D. They overlap with each other. Then the number of conformations that they can take in this overlap state is relatively less, because here the entropy is coming from the number of conformations. Now, each is trying to explore different configurations within half the cylinder, approximately. And if this was an infinite cylinder, if they segregated to two different halves of the cylinder, the number of conformations each can take is significantly more. So, number of configurations more means entropically it is favorable. It will increase the number of entropy. Yes, this is a thermal system, but the number of configurations is more. Hence, free energetically, it will be minimal if they are segregated along the long axis. So that is the hand waving way of understanding it. Of course, you can do calculations also. Now, so entropic segregation, that's how it would work. Two polymers would prefer under high confinement. High confinement is important because without that you won't see. It would have enough space to remain mixed or whatever. If you have a cubic box, yeah, there is no symmetry breaking direction. In which direction will it go? In a cylinder, there's a symmetry breaking direction. So here it will segregate to two different paths because there is confinement. Now, if the width of the cylinder was such that each could maintain its own size, right? They wouldn't get stretched out. They wouldn't have to fight for the number of conformations. Suppose these walls were very far, then you're not going to see this. But if the walls are such that they are confined, they're jostling for the number of conformations, then they would segregate. Now you can do your analytical calculation in infinite cylinders. In finite cylinders, you can't do analytical calculations. If this is like the aspect ratio is high enough, they would segregate, and that's what I showed you. Entropically, they would get larger number of configurations if they don't overlap. That was the content of the movie. However, if the cylinder becomes shorter and shorter, entropic segregation will not work anymore. Or if the cylinder diameter becomes higher, it won't work anymore. In fact, Subjun, the person who proposed this idea, entropic segregation, By biochemical means, he could actually increase the size, the diameter of the bacteria, giving some chemical. But rest of the thing was working fine. Rest of the cell was working fine. And there he saw explicitly, there's some series of 2012, 13, 2012, 2013, the higher diameter bacteria, chromosomes don't segregate as well. Okay. So these are some details of, so we took essentially 200 monomers, 200 beads per ring. We ensured that they were non-concatenated ring polymers. The cylinder diameter was 5A and length was 25A, 1:5 ratio. And this is important because later when we shall extend this work to bacterial chromosomes, we shall maintain, I mean, before it divides, the bacterial chromosomes, I mean, bacterial cell goes from 1:2.5 aspect ratio to 1:5. Here we are taking two full ring polymers, like two full daughter DNA. So we're maintaining the same length. Radius of gyration, which is estimate of the size of the ring polymer in free space. The diameter will be actually 14A, whereas the cylinder diameter is 5A, which means it's highly confined. And these are some blob theory calculations which can show you how will the free energy change with the diameter of the cylinder, but we are not going to discuss that. We are going to keep it qualitative because I'm trying to give you the picture. Okay, so this is the background. This is what is polymer physics. This is what is entropy. Why ring polymers segregate under confinement. I hope you get some point of idea. So after this, we're going to play. So this was basically work which I produced from previous work. Now we're going to play some games, and why we are playing some games will become obvious in a matter of 5 minutes. So we have been playing with ring polymers till now, but I shall make a topological modification. Topological modification means suppose we have a 100 monomer chain. Then we are going to connect by a spring diametrically opposite points of the ring polymer, okay, so that it is going to become an inverted 8. So you're going to have two different circles like this. And later you shall see we are going to also cross-link, introduce a spring between the 51st monomer and the 100th monomer. So this is a 200 monomer ring. So that we're going to have two small rings and one big ring, right? And this will become relevant. So you have two small loops and one big loop. So we call this the arc 1, 2 because there's one big loop and two small loops. Now, instead, so what I already told you, that if you take two ring polymers, confine them to a cylinder, they are going to segregate. Now, if each ring polymer has two smaller rings, yeah, because of this cross-link, so this is what we have put inside a cylinder. Then what you have, that of course the two ring polymers will segregate to two halves of the cylinder, but now you have two smaller rings inside each ring. And these guys, these smaller rings which are connected here, will also entropically repel each other, right? So not only this polymer repels this polymer, the smaller internal rings also repel each other. As a consequence of which, if you plot the center of mass of the monomers of this smaller ring, then it will be local and plot it. I mean, you plot the center of mass, the probability distribution of the center of mass of this ring, and plot it, and this x-axis is this length, this long axis, divided by the length of the cylinder, so that the length is 0 to 1. So then this guy gets localized, the center of mass gets localized here, the monomers get localized here. Normally a polymer is a completely disordered object, but under confinement, because of entropic repulsion, the monomers of this sit here, the monomers of this sit here. This is subloop 1, 2, 3, 4. The monomers of this sit here and the monomers of this sit here. But these two guys can also flip among each other. Hence, you see overlapping distribution of the probability distribution of center of mass. So you see blue and orange, subgroup 1 and 2 can interchange position. This data collected over many independent runs. And sometimes even polymers can interchange position. But all over and above what you see is basically that these loops segregate entropically and remain well separated along the length of the cylinder. Now, if you, from the center of mass of this loop, you draw a vector to the center of mass of this loop. So this is a vector and this is another vector from center of mass of this loop to the other, and take a dot product. Then and plot what are the cos thetas you are getting. What you see is you're getting a peak at -1 and a peak at close to 1, which means these two vectors either plot, point parallel to each other or anti-parallel to each other. They also have other angles, but the probability is less, which again is an indirect uh, proof that these loops stay well segregated along the length of the cylinder. And this is what we have, this is time or iterations in the simulation. This is the center of mass of one of the loops. This is this red color, the center of mass of the other loop, and so on and so forth. What you see is once in a while they interchange positions, but otherwise this remain well segregated along the length of the cylinder. So this is Z by L. This is Z by L. So it varies from 0 to 1 and they remain well segregated. When the green comes here, the red goes here. So this is what we are showing is this is well separated. Now let's play a little some other game. I'll finish my talk in 5-10 minutes. We're going to play with such a polymer. So this bunny, two small loops and a big loop. And you have two of such polymers. So each of this polymer is, you should think, so this polymer is sitting here, another copy of the polymer sitting here, red, but now you have two bunnies. Moreover, for reasons that you will soon figure out, there's a biological reason. So here you have 500 monomers. So first monomer is connected to the middle of the left arm or whatever, right arm. So middle of this arm, 125th monomer, there are 250 monomers in this arm and 250 monomers in this arm, 500 monomers in each polymer. So total you have 1000 monomers, 500 for this, 500 for this, right? And these are cross-linked. So you have these two bunnies in the middle of the arm. And if you connect the 250th monomer opposite end, which later, if you know the biology, it will be the ter, we also cross-link this. At the moment, I'm not giving any reason, but later it will have some consequences. And now we look how these different loops are organized. Then you'll see that basically, if you have this configuration and this high confinement, uh, these polymers will be organized like this. Two loops, these two small loops will have a tendency to overlap. So what I'm plotting is the probability distribution of the center of mass, COM, and uh, you know, and the center of mass of this, the center of mass of this loop 3, the loop 3 is here. And for the other polymer, which is denoted by stars, again, the big loop is here. So this is simulation data. And from this data, you can guess that this polymer is organized like this. So these two small loops entropically repel this big loop, which entropically repels this big loop. These two are connected. And this in turn repels both this loop and these two small loops. Now they can't flip because these are connected. The big loops have to be in the center. Hence, the two small loops are pushed to the poles of the cylinder. These are the two ends of the cylinder. So we have been talking about what is the position of the center of mass of the loops. What about the position of this particular monomer? This particular monomer is monomer number 1, which is now located here because we have made two loops out of this. Now, this guy is essentially sandwiched between these two small loops and the big loops. And similarly, The monomer number one from the other polymer is sandwiched between these two small loops and the big loop. These guys are repelling this. So this is stuck in the middle. So if you plot the probability distribution of monomer number one of each polymer, this is the monomer number one of each polymer, which after the topological modification has become something like this. Yeah. Then you will see that each of this is basically localized to one point. Normally, the polymer is a very random object. It keeps changing its configurations due to entropy, right? It can be all over the place. But because it is confined within the cylinder, you see it gets localized due to entropic considerations. I shall soon move to chromosomes, but now let's look at the position of some other monomers which are tagged. So suppose what about a monomer which is sitting here? Remember, there are always two polymers. Here I've shown only one polymer. Each polymer has this topology, this architecture, if you like. So this guy, R1, it will be pushed towards the end of the cylinder, right? And there will be two such polymers. So both the R1s occupy the end of the cylinder. What about L1? L1 is close to this monomer number one, but it's part of this loop. So it will be left. So if this monomer number one is here, this guy is pushed towards the pole. It's more towards the left. L2, which is a bit away, but part of the big loop. This guy will be basically more inside, So this will be L2. So this will be bit inside. And its probability distribution, plotting the probability distribution of each of these monomers, function of Z has been normalized. There are two polymers, so its length is 35 and diameter is 70, will be localized somewhere here. Yes, because of entropy, it can move to different points also. What about R3? R3 is part of the big loop, and there will be two R3s, one from here and one from the other polymer. And it is basically away from, it can be anywhere around the middle of the cylinder with low probabilities to go towards the end. What about this guy? This is stuck between the two polymers. So these guys preferentially occupy the middle of the cylinder. This was even known before. With this background in polymer physics, entropy, topological modification, localization of certain monomers in a completely disordered system due to confinement and entropic consideration, let's connect what you have learned back to chromosomes. So that's the third part. Now I'm going to link chromosomes and polymers and entropy. Why was I playing these silly games of figuring out why certain monomers are localized and where in space? Let's understand that. So what they do in experiments, so I'm again coming back to chromosomes before I link. So what they do, so this is basically, I think this is a picture from, yeah, Sigden's paper 2014. What they do is tag different monomers, base pairs or genes or segments of base pairs along the chromosome. So they tag it by different fluorescent molecules. And this is how it looks. You see, some are long, some have two red points, some have more red points, maybe you can, yeah. And what you see is these red points and the green points, different points in the chromosome, they take definitive motion. So you see this one is about to divide, there's a certain order to it, here there are two and so on and so forth. This is what they do. They follow the position of these tagged points in the chromosome under the microscope and see how the position changes as push the function of time along the long axis of the cylinder. 2 minutes of biology. I already told you that bacterial DNA is a ring polymer. Uh, Now, some of this might know, but I mean, the replication, making a copy of the mother DNA into two daughter DNAs, that's the process of replication. Originates, it starts at the origin, for short, ori. So this part of the chromosome becomes two parts. There are two copies of this. Yeah. So this is the mother DNA. These two parts are the two oris of daughter DNAs. It has made a copy of itself. Hence, there are two of these. And then replication proceeds along this arm and along this arm to terminate at the so-called ter position. So the replication is done by some protein machinery, which we call the replication fork. I mean, this position is called the replication fork. The protein machinery is called the repliosome, but you don't need to worry about it. This is mainly intended to be a physics talk. So what happens is this part of the mother has been copied. So this is daughter DNA1, this is daughter DNA2, or vice versa. So this is daughter, whereas this is mother. So this moves along this and this moves along this. As this moves, more and more of the mother DNA is being converted into daughter DNA1 and daughter DNA2. And replication will end here, where these two replication forks will meet at the third. And at the end of it, you will have two ring polymers. Of course, this is a schematic diagram. And the third will remains connected till the very end before cell division happens. So you have two ring polymers which look on two different parts, but actually the question is, how would they segregate? Right? How will they demix? And of course, it was suggested that entropy plays a huge role, and this figure is from the Jongren paper, again from Genes and Development. And just before cell division, this third connection gets cut. You saw previously I had attached the two ring polymers at a certain point, and that was the ter, and had also marked it as the ter. Now, some more quantitative data. Now, this data is by Kaas et al. Biophysical Journal 2016. Now, what this guy had done is tagged 7 different points along the chromosome contour. And what is shown here is an average over 10,000 cells. This white part is the length of the cell. It gradually grows to double its length. The ori is the point where replication begins. It's typically seen to be at the middle of the cell. So here replication has started, but you can't distinguish the two oris. So it remains in the middle of the cell for around 20 minutes. And after 20 minutes, and this red line is the standard deviation in the position of the two oris. You can't distinguish them. After around 20 minutes, something happens, and the two oris segregate and go to two different halves of the cell. So the ori from the center, the two oris from the center segregate to go to two different parts of the cell. And from the center, the half, it goes to the 2/4 position. So this is like the 1/4. So from here, it is the 1/4 position and the 2/4 position, and it remains there. After that, cell division happens, after so on and so forth, right? Not only that, if you look at the other tagged points, so now this is time, this is the length of the cell, the long axis of the cell. What is being shown is the ori is in the middle. But then at a certain point in time, the two oris separate out and go and sit at the 2/4 position. Some other point L2, which is tagged here, initially it doesn't have a particular point. From the middle of the cell at a certain point in time, it goes and sits not quite at the quarter position, a bit inside of the quarter position. Some other point R3, well, here it's in the middle, but then it's all over the place in the middle of the cell. The ter initially is delocalized, but towards the end, it goes and sits in the middle of the cell. How does this happen? So what we propose is essentially the chromosome has this topology, and due to this, note previously when I was talking about two full ring polymers, but now there is a replicating chromosome. So initially you have one mother chromosome like this, and if you have one mother chromosome like this, what happens is the two loops. So you don't have two ring polymers, you have only one polymer. We suggest that the chromosome, so this is our assumption, this is our hypothesis, uh, that if the chromosome actually occupies is has this topology, the mother chromosome, then you know the ori will be in the middle. Now, even as this is the original part, this is why the ori is in the middle. Now, as replication is happening, each of these groups are being made a copy of. And how am I modeling replication? I won't tell you for the sake of time. But as replication happens, this architecture, mother chromosome, there's more and more chain being added, the two daughter DNAs being added. And when there is enough replication, essentially what happens is we are claiming is basically each of the two daughter chromosomes takes the conformation of this arc 1, 2, these two small loops and the bunny. And when that happens, you already know that this monomer 1, now you can note that I had actually marked this OEC and this was stirred and so on and so forth. These would get localized to the quarter position, right? And when will these extra loops be? So initially the ori is in the middle because there's the mother, but as replication proceeds, as there's more and more daughter DNA produced, when half of this is produced, when half of this has become daughter DNA, then you put in the crosslinks, there will be increase in entropic repulsion because now you have these small loops. Of course, the mother is still just one loop. This has been replicated, but then it is then that the ori will go and sit from the middle position to the two quarter position. Now, in hindsight, you see, I had marked them R1, L1, L2, R2, R3, and these are exactly the same convention as was done in the experiments. And the principles of localization, which I was explaining, R2 is, localized a bit in the inside, and R3 is all over the place. That is the principle that I was explaining previously. And ter comes in the middle, and so on and so forth. Okay. In hindsight, I told you that this segregation happens, movement of the two oris, separation happens after 20 to 25 minutes. Later we realized that 20 to 20, the full time to have the replication for the E.coli chromosome actually takes 40 to 45 minutes. And 20 to 25 minutes is exactly half of 40 to 45 minutes. That's the time when this part of the chromosome and this part of the chromosome has been replicated, and you will have this excess cross-link because each daughter will take this new topology, and that is the time where the two oris will segregate out to two different halves of the cell, right? So everything fits together. So that was our proposal, but there's no reason that you should believe us. And in fact, when we did our result and explained all of this, we didn't believe. We went to biologists and said, do you believe us? And they helped us. We not only matched all these localization data, but in hindsight, we could predict the time or the stage of replication. And there are many other experimental data, which I'm not going to talk about because it will take too long. There are a lot of things which we matched wonderfully. So what I'm going to do here is show you a movie. So here there is initially the blue chromosome is the mother chromosome. Okay. So let me just start the movie first. What you are going to see is there is a cylinder. Just let me stop it. So what you see here is there's a blue chain which has the topology of that bunny, two small loops and one big loop. And the ORI, the red molecule, the red big spheres are ORIs. It is made big only so that you can identify, and they're roughly in the middle of the cell. Yeah. Now there is being replication happening, which means I'm adding more and more monomers to it to make the daughters and the daughter DNA1 and daughter DNA2. Now, I am doing a polymer simulation, bead spring polymer. There's no protein, there's nothing living in that. All that I'm doing is maintaining the topology, maintaining the crosslinks, right? And if a daughter DNA, so if the mother goes into daughter DNA1 and DNA2, in terms of bead spring model, there's hardly any differentiation between daughter DNA1 and the mother. They're just chains. But except that more and more chains of daughter DNA2, monomers of daughter DNA2 is being added. So there's no distinguishing factor between daughter DNA1 and mother. So here we just keep it blue. But you will see that pink monomers are being added corresponding to daughter DNA2. And as you will see, the length of the cylinder, so let me now. So this yellow is the tur, it is all over the place. The red two spheres are roughly towards the center of the sphere. You can observe that the cylinder is increasing in length. So its aspect ratio will go from 1 is to 2.5, 1 is to 2.5 to 1 is to 5. And you'll see that the ORIs are basically fluctuating somewhere in the middle here, and pink monomers daughter DNA2 is being added. You can't distinguish between daughter DNA1 and daughter DNA2. Essentially, the replication forks are moving. At a certain point, you will see that the two red balls will go to two different halves of the cylinder and the yellow will come in the middle. And simultaneously, now you will basically see the yellow has come in the middle. The two ODs have moved and moved from the middle to the average quarter position. And simultaneously, the polymers have segregated to two halves of the cylinder. And that is exactly what you see in experiments. By the way, these movies are there also uh, on my web page. So let me and so all that I want to say is, initially the ORI was somewhere in the middle of the cylinder. Here, the cylinder has become much more elongated. What I have plotted is the position of the ORI, the two ORIs, and they are basically uh, localized near the quarter positions. This red line is with the topology of arc 1, 2 small loops and one big loop. On the other hand, if you have just the ring polymer, no modified topology, ORIs can be anywhere. This is called this arc 0. Arc 0 corresponds to the ring polymer. The ORI can be anywhere along the length of the long axis. You don't see any localization. You don't see any localization even when replication has taken place, except that one ring polymer has become two ring polymers. You see localization of the ORI, localization of the arc, only if the ring polymer adopts the arc 1, 2 or arc 2. I mean, these two small loops and one big loop. Other than this, we matched many of the experimental details. I can hypothesize anything. Any theorist comes and gives a model, an explanation. You should say, why should you believe? Why should I believe you? How much, what have you masked? Do experimentalists take you seriously? So the answer is yes, we have. In our papers, we have put the experimental data and the theory data next to each other saying that, this is what we have managed to reproduce, this is what we are trying to explain. And other than what I'm saying here, we've explained other quantitative data. Yes, this is a spherical cow model. Now biologists have come to us and asked, okay, that's very good, but can you explain more such data? So now we are adding complications to the model, because in reality, DNA is going to have much more loops than just two small loops, right? So I'm adding small loops and trying to explain other intricate details of the simulation. This is biological data. This is from our simulations where different points essentially segregate time with relative to origin split. Uh, they have nice, smooth, thin lines because their data is averaged over 10,000 cells. We have 50 independent runs corresponding to 50 cells. So we have much more statistical fluctuation. Yeah, these are Monte Carlo simulations, not exact. Yeah, so slightly different from Lajwa, but it has the same principle. Other than that, I'm not going to talk in detail. Introduction of these smaller loops actually increases the entropic forces of repulsion. So we studied a large number of topologies and figured out, the one question was, is there enough entropic force to segregate the two halves of the DNA? Sorry, the two DNAs to two halves of the cell. We have seen that adding these smaller loops actually increases the entropic repulsion that leads to faster segregation. Some more work is going on around in the group. This is where I would like to end. So most of this work was done by Debarshi Mitra, who was my previous PhD student. Tejal Agarwal was the previous PhD student who actually went to Sukhjunjun, learned about the experiments, and before finishing her PhD, she came to me and said, there's an open problem if you want to look at it. And that is the problem I gave to Debarshi Mitra, who solved the problem. And this is my current PhD student. I haven't talked about his work, but let me very briefly tell you, this matched very fine with experiments, whatever I told you. Uh, let me let me tell you about a conundrum. Okay, so what I showed is basically one mother chromosome becoming two daughter chromosomes, segregating to two halves of the cell, and cell division takes place. And I know nothing about cell division. I told you that replication takes 40 to 45 minutes. There's no way, because to do replication, these chromosomes have to be opened up by some helicase molecule, and that can rotate at a maximum rate of some 30 rotations per second, and so on and so forth. So replication cannot happen faster than 40-45 minutes. You can slow it down if you want. You can add some chemicals and slow it down, but you can't do it faster than that. But if you give the cell good food, whatever good food means, maybe the biologists among you might know, but there is this minimal media and protein rich media, then one can see the E. coli cell divide into two in 20 minutes, 30 minutes, right? And then the two daughter cells will divide into two granddaughter cells in another 20 minutes. So one cell to two cell in 20 minutes, two cells to four cells in 40 minutes, in another 20 minutes, four cells to eight cells in another 20 minutes, and so on and so forth. Right? Perfectly healthy cells. But I told you chromosomes cannot be replicated in less than 20 minutes. So I hope you realize that there is some contradiction. There's something which doesn't match. If replication is not complete, how can a new cell be born? Right? That seems a contradiction. This contradiction was actually solved much before me in the 1960s. Basically, when an E. coli cell senses that there is good food, somehow it does something to its machinery. So even before one round of replication is complete, another round of replication begins. So basically, while the mother is still getting replicated to the two daughter DNAs, and there is part two daughter DNA chromosomes, the two daughter DNA chromosomes, they have the two origins, they start getting replicated and start producing granddaughter DNA. So while the replication of the mother is completed and and you have two daughter DNAs, half of the, I mean, some part of the daughter chromosomes have already been converted into granddaughter chromosomes. So granddaughter cells, two daughter cells are born with granddaughter DNA. In So newly born daughter cells already have 50% of granddaughter DNA. And similarly, another round of replication. So while granddaughter DNA, so the daughter becomes fully granddaughter DNA, the granddaughter DNAs would start another round of replication to create granddaughter DNA, and so on and so forth. So that's the biology problem. Where do we come in? We come in because now multiple rounds of segregation of chromosomes, these granddaughter chromosomes should not get entanglement. No, there's some entanglement released by topoisomerase, which we have also modeled, mimicked in our simulations. We have not talked in detail about that, else the polymers will remain entangled. But all this segregation and organization will need to happen now in multiple rounds of chromosomes, because you have granddaughter chromosomes, daughter chromosomes, mother chromosomes. This is very complicated stuff. And you have quantitative data. They have tagged different points on the chromosome, except some are now on the granddaughter chromosome, some is on the daughter chromosome, and there is quantitative data of that. And with our simple entropic model, we can match 80% of the data. That is the experimental data of Sukjun actually. So Cas had the slow growing and this fast growing bacteria with multiple rounds of replication going on in parallel, and quantitative data of how it segregates, where the different points, the loci of the DNA are along the long axis. Those quantitative experimental data, and that is what was basically modeled and simulated by Srirang Pandey. And we have a very long 23-page paper with experimental data and simulation data next to each other, published just now in 2024. Okay. So we saw that topologically modified polymers have some very interesting properties. So some other stuff was done by a previous IISER Pune BSMS student. I won't talk about his work. It's pretty interesting, but I shall just briefly mention an IISER Behrampur student, Kinkini, who is currently my PhD student, joined our group. But she did her MS thesis in IISER Pune. And after doing the bacterial chromosome, the obvious question which occurred to me is what would happen for eukaryotes with multiple chromosomes inside the nucleus? Nucleus is spherical, it's not cylindrical. Now that's too difficult a question to handle for me. But what Kinkini worked on, so she just passed out of IISER Bahrampur. What she worked out is basically topologically modified polymers inside the sphere, not only one polymer, no replication, but multiple polymers with various topological small loops and big loops inside a sphere. And the point that she saw is if you put multiple polymers inside the sphere, the smaller loops go and organize themselves and occupy the periphery of the sphere. Now, what will it be its biological significance? We are not yet up to it. I'm not going to discuss that. With this, I shall end, a pretty long talk. The basic message is we have used modified topology and entropic forces to localize polymer segments along the cylinder. We can control entropic forces of segregation by modifying the topology. I've not talked about this and I've not talked about this, but I've talked about slow growth and fast growth. I've got funding. We have got John Fulbright, and we have some biologists coming and talking to us, teaching us some biology because I knew nothing of this. I've gone to France and discussed with many people. I've been to a biology conference, bacterial chromosome conference in Israel before all this war. Uh, broke out, and have been mentored and taught the biology, discussed with many other people, and I thank them for all that they have taught us. My students have been wonderful. Without them, I couldn't have done this work. They also helped me teach biology, and I thank you for your attention. And I'm sorry for this extremely, relatively long talk. Okay, so bye, and thanks for your attention if some of you are still listening.